I'd like to talk about aphid community, symbionts, and barcodes. And I want to talk about some research that's been done in my group over the last 15 years or so, which has sort of spanned the introduction of barcodes. And we use barcodes a lot now. We didn't at the beginning. And I want to talk about some of the results that we've come up with, and then talk about how we've incorporated barcodes and what difference they've made and what difference they might make in the future. So we are motivated by both ecological and evolutionary questions, and especially in insect community ecology, we're very interested in how communities are put together, and especially communities of plant-feeding insects. Communities of plant-feeding insects, where most insects feed on different host plant species, and so many of the classic competitive effects that we think structure other communities are unlikely to be very prevalent in these communities. But they share many natural enemies, and one of the things we've been particularly interested about is how the natural enemies, how the next trophic level can structure the community through apparent competition. So this is actually a leaf miner community here, linked by their shared parasitoids. And so we build food webs in order to try and understand how they're structured, and in order to build experiments. So for example, we might come up with a hypothesis about how removing one of the blue species might affect the dynamics of the other species through a shared natural enemy. But in this talk, I'm largely going to be talking about aphids and the community of aphids that we've been studying in the southeast of England. So not perhaps the most charismatic of all animals, but they're wonderful to those of us who study them. But even more wonderful than aphids are the insects that attack them, and especially the parasitic wasps, the group of organisms I love the most. These are some of the parasitic wasps that attack aphids. In addition to the direct parasitoids, there's a guild of hyperparasitoids that attack them as well. Aphids suffer depredations of parasitoids, but also of a group of fungal entomopathogens as well, much more poorly known taxonomically. And in addition to that, they're attacked by groups of predators, many of which you'll know well, including lacewings, including hoverflies and ladybirds, many of the very familiar insects that we all know very well. So what we've been doing is, over actually many years, constructing food webs of this community. So for example, this is these are quantitative food webs showing on the lower register the aphids. The width of each bar is proportional to the abundance of that aphid species, and then there are parasitoids and hyperparasitoids above it. And so you can take a complex food web time series like this, which is a statistical nightmare to analyze, and try and make predictions about how the different components interact. So that's sort of the time dimension. We're also very interested in the effects of the different guilds of predators. So for example, on the top, predators are far harder to study than parasitoids. When a parasitoid kills an aphid, then it sort of leaves the body in the library that you can come and see who's killed what. It's much harder with predators. You've actually got to watch who eats what. And so you can see that there are predators, which is a highly connected web. Most predators attack multiple aphid species. The parasitoids, the primary parasitoids on the bottom, are much more specific, as are the fungal pathogens on the lower left. And the parasitoids, and especially a group of what we call mummy parasitoids, are much more polyphagous. So we think that it is at these levels where we have natural enemies that feed on many different alternative prey items, that the structuring force of apparent competition is strongest. And so we have a sort of hypothesis for our community that there are three structuring sort of modules within it. At the bottom, there are the plants, and the plants are obviously competing, but we're entomologists, so we don't really worry too much about that. At the next level, we hypothesize that the way that the aphids feeding on different host plants interact the most is through shared predators. And in a moment, I'm going to show you some experiments in which we've gone out to explore that. And then I won't show you about what happens at the next level. The next level is slightly more complicated because there are these two groups of secondary parasitoids. But we think, and we have some experimental evidence, that shared mummy parasitoids, parasitoids that attack aphids after they've been mummified by the primary parasitoids, that they have an organizing role there. So just to give you a couple of examples of experiments with predators, and this is work done by the late Christine Muller. So the type of thing we do is we take a aphid and we explore its population dynamics in the presence or absence of other species. So the blue control line there is a typical trajectory of an aphid population in the temperate region. This is a period through from about the middle of June into about now. And you can see a typical a typical aphid population trajectory there, the control line. In the case of the brown line, what we did is experimentally we put in the vicinity of our focal aphid another species which we know is a very good source for the coccinellidae, for the ladybirds that attack him. And so we're having an indirect interaction here. The presence of an aphid feeding on the different host plant is having quite a major effect on the population dynamics of our focal aphid species. And let me give you an example of another experiment we've done. One of the nice things about aphids is that you can actually ask questions about what species are not present in a community. So you get a suction trap, which is essentially a domestic hoover lying on its back, and it sucks aphids out of the air. And so you can identify these aphids. When we did it laboriously using keys. Today we'd use barcodes. You can identify what aphids are coming down in the aerial plankton and then say, well, why is that aphid not there, even though it's sort of raining down into our community? And one such aphid that we expected to be present was this thing called Brachycaudus cardui, which feeds on Senecio. You can see the picture up left. And so we initiated our own colonies to try and see what was keeping it out. And what we found was there was sort of diffuse apparent competition keeping it out. So the red line there is if you just put a colony on one of the Senecios and it quickly dies out. We then protected it from the typical carabid beetles, the generalist ground predators that climb up all plants. And the colonies lasted a little bit longer, but they still went extinct. And it was only when we gave it total predator protection, in particular from the aphid specialist predators that were maintained by the other aphids within the community, that we were able to get the aphid to establish. So this aphid species was being excluded by diffuse apparent competition. And interestingly, in our study site, ants are quite rare because it's a rather damp site. But if you provided, in a fourth treatment, if you provided ants by putting colonies in flower pots with sort of string going to our plants so they could come across, then you got exactly the same protection as if you artificially prevented predators from getting at it. So at the time that we were studying these community effects, we were also interested in evolutionary questions, especially the evolution of resistance against natural enemies. And we've been doing a lot of work on Drosophila, and prompted by some work actually by Heather Henter, who I haven't seen for ages, and it's delightful to see her at this meeting again, to look at some of the relationships between aphids and their natural enemies. And in particular, we looked at the pea aphid, which is a sort of Drosophila melanogaster of the pea aphid world. And one of the nice things about aphids is that you can freeze a genotype, and you can look at a particular genotype, because aphids, as all of you know, are clonal during the summer. And so what I've plotted here is the susceptibility of different aphid clones to two species of parasitoid. Those are Aervi and Aida. Sorry, I should have written them out. And a fungus at the bottom. And so you can see that there is substantial clonal variation in resistance against these guilds of natural enemy. And Heather, who was then working with Sarah Vere, had some evidence that there might be a trade-off between the two. And we did a whole load of experiments, and we didn't find that. And we just got a whole series of confusing results, which in the end we wrote up for Evolution. And I thoroughly advise you never to look at that paper, partly because it's confusing, and partly because just after it came out, a group from Tucson, Kerry Oliver, Jake Russell, Nancy Moran, and Molly Hunter, came up with this really interesting result, which no one had thought. So about that time, it became apparent that many aphids, in addition to their obligate symbiont, so all aphids have a symbiont which they need to make the amino acids that are absent in their phloem diet. In addition to their obligate symbiont, many of them had facultative symbionts. So one clone will have one, another clone will have another. And Molly's group found that one of them, named Hamiltonella after the wonderful Bill Hamilton, Hamiltonella conferred resistance against parasitoids to the aphids that it was carried in. And this instantly made sense of a lot of the results that we had that were essentially had baffled us beforehand. And we, as I said, had also been studying the fungi, and so we immediately looked at a really curious result that we had. So on the left are 10 clones from a species of birdfoot trephile, all of which are highly susceptible to this fungus Erinia. And on the right are 10 clones from clover, and that's not a PowerPoint glitch. All of them were resistant against the fungal pathogen. And it turned out that when we did some molecular biology, that all of them carried another symbiont, not Hamiltonella, but a symbiont called Regiella, actually named after a wonderful insect physiologist who many of you will know, Reg Chapman. So this was highly indicative. And what we then did was we did an experiment. And again, one of the nice things about this system is that you can manipulate the presence of the symbionts. You can cure them using specific antibiotics, and you can inject them back in. And so what we did is we took 5 clones of aphids there, the codes on the bottom, and we looked at the survival with and without symbiont. And you can see that with the symbiont, they survive reasonably well. In our particular experimental protocol, there is about 40%. If you remove the symbiont, the survival drops very, very strongly, down to about 5%. If you don't believe the results on the right, look at the results on the left. They'll convince you. So it looks like that a different species of secondary symbiont protects against a different natural enemy. And there's a nice twist to this as well. Again, as many of you know, aphids live in either very dense colonies or, in the case of the pea aphid, in rather looser colonies. So if you're killed by one of by the fungi and you become this sort of sporulating mummy, then what happens is that you infect your co-clone mates. So even if you're going to die from this fungi, you can still increase the inclusive fitness of your clone as if you die but don't sporulate. And so that we found that with the symbiont, sporulation frequency also went down. So even if you're killed, if you've carried the symbiont, you sporulate with lower frequency, you're less likely to infect your co-clone mates. And of course, those co-clone mates are carrying the symbiont as well. So fast forwarding to today, we now know that there's a whole menagerie of symbionts infecting aphids out there. That's the name of the primary symbiont, which is essentially an organelle. Its phylogeny is exactly congruent with that of the host. And then we have this zoo of other secondaries, including Hamiltonella, which is the one that Molly's lab showed protected against parasitoids, and Regiella that we showed protected against fungi. And we're still trying to investigate what the others do. Most of them are Enterobacteriaceae, but they map onto different parts of the prokaryote life cycle as well. Let me give you 2 examples of recent research from my group. So we've been systematically looking at whether the other symbionts have effects on resistance against fungi, and we are finding it. So if we just look at the top one, which is survival, in the protocol is slightly different here. So it's the experimental protocol can tell apart the effects of the symbiont a bit better. If you have no symbiont, then you essentially die from the fungi. But a whole series of symbionts give protection, although not Hamiltonella. Hamiltonella on the right, that's the parasitoid one. And then within spiroplasma, then we have some strains that give protection, some don't. What we're trying to do at the moment is to sequence all these different symbionts to see whether there are sort of cassettes in common, whether there's a common mechanism. We still don't understand the mechanism of protection against fungi. We've also been working on some of the parasitoids as well. So here are the members of the two main groups of parasitoids that attack aphids, the braconids, the Aphids at the top, and this cute little Aphilinus at the bottom. And recently, Elsa McLean in my lab has found some interesting results. So we have strains of the Hamiltonella that we have collected from aphids that are feeding on rest harrow and Nonus at the top, and essentially they have little effect on proportion parasitism. So that level, sort of 75 or 90% escaping, it makes very little difference. But then we have some strains which are very good at protecting against Aphilinus and poor against Aphids, and the other way around. And interestingly, these strains are correlated with the food plant upon which the aphid feeds on. And this is just to summarize on the symbionts and the natural enemies there. So we now know that many different symbionts have effects on parasitoids and fungi, and work in particular from Kerry Oliver has actually found out how it works. It involves phages that deliver eukaryote toxins. And then we have different symbiont species can protect against the same natural enemy, and then we have this variation within symbiont species that is associated with host plants. And this is the other bit of the complexity of the pea aphid. The pea aphid consists of a series of races that are specialized to various extents on different host plants. Some might actually be different enough to be full species. Others are much more similar. And we've been exploring using a big panel of 1,100 aphid clones collected from many host plants all around the world exactly how the aphid phylogeny matches onto host plant use. If you do a phylogeny, actually this is based on the Buchnerer gene, then you find that there is some conservatism of host plant use, but there is some mixture. It appears that the genes that enable you to use a particular host plant, there are sort of small number of cassettes that can introgress into different groups. If you take the four most common symbiont types, and these are haplotype maps where the different colors are different host plants, you find substantial structuring due to host plant use. And again, what you can do is you can look at how the phylogeny of the aphid and the phylogeny of the symbiont, how they interact there. And that's one for Hamiltonella, and that's one for Regiella. In Regiella, you can see particularly this association with the biotype feeding on clover. And then you can do some of the analysis. You can do experiments, so you can try and shift the symbiont composition and see if it affects host plant use. But curiously, while you get very clear results when you do equivalent experiments for natural enemies, it's much more complicated with host plants. But what we found, and this is work from Lee Henry, is that using ancestral state reconstruction, we find that if you acquire particular symbionts, then it increases your probability of colonizing, of being associated with a new host plant. And clearly, this is a correlational study, so you have to be cautious about causation. But it does look that if you acquire Hamiltonella, you're more likely to colonize trefoil and rest harrow. And if you acquire one of the two main clades of Regiella, you're more likely to colonize trifolium. And the last part there is that there is two symbionts which seem to synergistically work together to protect against certain types of parasitism. And quite nicely, we were able to pick out that synergy from the phylogeny as well. The other point that comes from this work is that the major clades are quite old, much older than host plant races. And, something's gone wrong here, never mind. And so it immediately makes you wonder what happens if you go from pea aphid to the whole community of aphids. and that is a PowerPoint glitch, but you can see there are a little over 100 aphid species, and we use barcoding for this around the edge. And you can see the association of the major symbionts. And there is some major phylogenetic structure in that. A technical thing is that most of what we know about symbionts come from the pea aphids, so an immediate question is, are there some other things out there, or are the primers that you've developed from the symbionts that attack the pea aphids, will they work in other parts of the clade? And so we've done some next-gen sequencing to look at that. And essentially, we think we've discovered most of the major symbionts, at least in the aphid clades that we've looked at in Europe. And let me give you one example of a result coming out of that. I'm rather frightened to talk about insect-ant association speaking just before Naomi Pierce, so I hope she won't find problems with this. So what we looked is the probability of a species being infected by Hamiltonella and Regiella, depending on whether they were a species that was typically tended or not tended by ants. And the probability, the significance test in there, takes into account all the complex phylogenetic effects. And what we find is that if you're not tended by ants, you're more likely to carry Hamiltonella, whereas there was no effect for Regiella. So what we think here is that aphids can have alternative mutualists to help defend them from parasitoids, either ants or the symbiont. So just to summarize this, it's amazing. Aphids are the major pest of temperate agriculture, probably the single most important pest of temperate agriculture. And much of what we thought we knew about their biology 15 years ago, we're far less certain about today. Symbionts are much more important, certainly in aphids. And I think increasingly we're finding that in other eukaryote systems. Secondary symbionts, they provide a reservoir of adaptations that the aphids can sort of sample from. Our sort of working model on this is that it's a bit like bacteria sampling plasmids from different communities. So they're a sort of horizontal gene pool, rather like the plasmids are a horizontal gene pool. And then I've just mentioned there's evidence of alternative defense systems. So last couple of slides. When we started off, as I said, we didn't use barcodes at all. And I think one of the questions that we ask ourselves now is that if we had better taxonomic identifications, would it have affected that some of the food web metrics that we've used to compare food webs in the past? And I've showed you 2 experiments that haven't worked. I haven't showed you the 50 or 60 that didn't work. If we had that, it wasn't quite that many, just before those of you doing Bonferroni corrections in your head. If we had better taxonomic information, would we have designed those experiments to look at apparent competition better? Now, I'm slightly comforted this by work that Thomas Rosling has done, who's taken, in fact, some of the leaf miner communities that we've studied as well, and he's found that, at least in the well-studied fauna in Europe, the morphological taxonomy is pretty good. And I think the real thing that I would like to see as a community ecologist is this continuing trend towards robotic, cheap, really high throughput barcoding so that we can identify vast numbers of specimens very quickly. A lot of the work I've shown you on the symbiont structure and the pea aphid structure has been sort of below barcode resolution, and so we've used multi-locus sequence typing techniques. The aphid symbiont community structure, we could have done it without barcoding in the UK, where essentially every species has been studied by a Victorian vicar, but it would have been much, much hard work. And I think if you're doing it in the less well-known fauna, much, much harder. And I finished with an old hobby horse that I've gone on before, and last time you were kind enough to invite me to Guelph, you asked me to talk about this. So barcoding and many of the other molecular techniques are going to render obsolete many of the sort of craft skills of taxonomy, building phylogenies, doing identifications and things. But a barcode bin is valuable, and you can do some sort of macroecological things. But it's of limited use. It's even of limited use if you have a Linnaean name to it. And so I just think there's a real need for taxonomy to reinvent itself as a sort of custodian of the information space, as a science of biodiversity. And so my last slide is two things. First of all, to thank the organizers for inviting me here, and to say what I hope will happen in the next 10 years is that we take these sort of bare phylogenies and bin numbers and things which are hugely, hugely useful. And we've... Okay, I started off by disgracing myself, and I finished by disgracing myself, which are hugely, hugely useful, but we need to populate them with this rich biological knowledge, and hopefully this will work. So we want to transform the bare phylogeny into the rich biological phylogeny. Thank you very much.
We have about 4 minutes for questions. For Charles, any questions? There's.
Hello, my question is that did you find any symbiont which would deter the hyperparasitoids? Because that would be much more important, particularly in case of Aphids, you know that mid-season hyperparasitoids take over the primary parasitoids. So I think that if there is a symbiont which deters the hyperparasitoids, it is much more meaningful in the field.
We have a paper which is, we're just writing up at the moment, and we've done it with one group of the hyperparasitoids, the alexistine chiripids, and we found no effect for that. We don't think there'll be effect on the hyperparasitoids that attack the mummy, because by that time it's a sort of dead mass of aphid. But that's a neat question, and we have looked at that quite recently.
More questions?
So you've provided a great example of the way that barcoding can enrich an ecological study and vice versa. How do we operationally and pragmatically better cross the gap between the ecological community today and the barcoding community? How do we increase those synergies?
We as biologists are spoilt these days by the resources that we can access from our desktop computers, especially the molecular resources. I think that in order to get the ecological and evolutionary community really lining up behind Paul's wonderful speech on the first day, that one needs to be able to present the wonderful summation of 250 years taxonomy in a form that's much, much easier to access. And I think until that happens, until an ecologist can, and we're spoiled, we shouldn't be doing this, until we can get that information, it's going to be hard for that community to be really arguing for many of the things that you want. So, and again, forgive me, this is a rusty needles, I talked about it before, these really rich web-based taxonomy resources which allow someone from the outside to access all that corpus of knowledge.
Hi, you said some of the symbionts, protect aphids from the fungus and natural enemies. Can you comment on with respect to pest management strategies? This problem in pest management strategies.
Yes. For the majority of aphid problems in temperate, high-input agriculture, I'm unclear whether there is direct implications for it. In some of the greenhouse systems where one is trying to do a highly biological control, then I think it would be helpful for you to know the symbiont status of the major pest. Now, actually, in those circumstances, the major pest is Mysis persicae, the peach potato aphid. And curiously, it's a very small, horrible aphid. Curiously, that has a lower frequency of symbionts than any of the bigger ones that we've looked at. We're exploring some of the relationships both with virus transfer and with insecticide resistance at the moment. But so far, I'd be sort of fibbing if I said that we've come up with some really important applied conclusions.
Thank you so much.
Paul.
So aphids are interesting organisms that are just flooded with energy. All of these carbohydrates, they just don't know quite what to do with it, so they leak it, and they seem to be quite an attractive magnet for symbiotic organisms of one sort or another. So I'm quite interested, you mentioned a little bit about placing this in a broader phylogenetic context. How many other organisms have tissues that are flooded with equally protective organisms, do you think? I mean, when I think about encountering a flu or a bacterium, I'm not necessarily thinking about my endosymbionts protecting me from it. So the question is, how broad is this pattern? Is it something to do with the fact that the breeding system of aphids, this clonal selection mechanism? Thank you.
Taking your last question first, there's a sort of control for that because one can look at other homopterans which don't have that breeding system. And so the whiteflies and some of the other bugs, they're chock full of symbionts as well. I think that in insects that feed on nutritionally unbalanced food, so you're right, it's full of carbohydrates, poor proteins, low in amino acids, then they tend to have obligate symbionts, as do, for example, blood feeders. And I think that they are pre-adapted to host these facultative communities. So I think they are more common on those groups that also have obligate symbionts. I think when groups have begun to look at the gut symbionts of other things, for example, ants, and Naomi will know much more about this than me, then I think they're finding lots of interesting interactions there. And then we only have to look at Wolbachia. So Wolbachia, which is present in a very large, significant fraction of organisms, and has this bizarre set of effects. That, to me, suggests that there is a lot out there. And one of the exciting things about next-generation sequencing is that we're getting to a stage where, essentially, you don't have to have that happenstance of finding a Wolbachia or a Cardinium and then finding it everywhere. We can now systematically search a group and see, well, is there any sort of smoking gun symbiont there that we may have overlooked?
Thanks.
Thank you.